Let's make a start. So good afternoon and thanks everyone for joining us and welcome to Alexandra Heller's Master's Thesis Defence. I'm going to start with a few introductions and then a little bit of housekeeping before handing over to Ali. So for those of you who don't know me, I'm Nicholas Webb. I'm Research Associate Professor at the Department of Plant and Environmental Sciences and the Jornada Experimental Range, and I'm serving as Ali's committee chair. On Ali's committee, we also have Dr. Brandon Bestelmeyer, research ecologist and supervisory research scientist at the ARS Range Management Research Unit and Jornada Experimental Range. Dr. Colby Brungard, who's assistant professor in plant and environmental sciences, and Dr. Akasha Feist, who's assistant professor in animal and range science. And Akasha is serving as the Dean's representative for this defence. So this afternoon, Ali's going to be presenting her master's research on an inductive approach to describing ecological dynamics with standardised monitoring data for the Rio Grande del Norte National Monument. A bit of background to Ali. Ali grew up in Maine and moved to New Mexico to attend St. John's College in 2009. And she became interested in rangeland ecology during a job training wild horses and burros for the BLM and Forest Service, and also learning about ecological and social contexts that drive land management. So Ali graduated with a bachelor's in environmental science from Prescott College in 2015. And has done extensive field work throughout the southwestern and southeastern US. And prior to starting her master's with us at the Jornada in 2018, Ali spent 3 years working for the Taos BLM field office, implementing their assessment, inventory and monitoring, or AIM program that you'll hear a bit more about today. So I'll shortly hand over to Ali for a presentation, and that will last about 30 to 40 minutes. During her presentation, if you can keep your microphones muted and your videos off, that would be great. Thank you. After Ali's presentation, we'll have the remainder of the hour available for questions. So we'll wrap up at 3 p.m. Mountain Time. And if you'd like to ask Ali any questions about her work, if you can notify me through the messages, so you can click on the chat icon at the bottom of your screens and send a message directly to me so that it doesn't go to everyone. That would be good. And I'll then call on you to ask your questions in the order that I receive them. And at the end of the hour, we'll wrap up the discussion so that Ali and her committee can move on to the closed defence. So thank you, Ali. I'll hand over to you.
All right, thank you, Nick, for your introduction, and thank you all for joining us as I talk through my master's project. So as I move through this presentation, I will describe what ecological sites are and why they're important for land management. I'll introduce the study area and the data set used. I'll review the methods and results and go over some implications, conclusions, and potential future directions. Ecological site descriptions are a land management and interpretation tool, and they divide the landscape into conceptual units based on abiotic properties, such as climate, geology, soils, landform, and hydrology, all of which vary spatially across landscapes. So in this example, we have shallow rocky soils where water tends to run off on the mountainous landforms. The lowlands have deeper soils with more clay where water tends to gather. Some variable soil depths and hydrology in the hills. And so each of these sets of abiotic conditions are able to support unique vegetation communities and ecological dynamics. So for example, the shallow soils on the mountains can support mixed conifer forest versus more grasses and shrubs on the flatter landforms where soils are deeper. So ecological site descriptions describe abiotic conditions and plant communities as they vary together spatially. And state and transition models are a part of ecological site descriptions. They organize information about how plant communities change over time in response to disturbances, climate, restoration, and land use. And the way that plant communities respond varies by ecological site. So we see an example state and transition model on the right. They are typically represented by these box and arrow diagrams where each large box represents a state. And the states represent consequential shifts in ecosystem structure and function. So we see the arrows between states, and they describe the mechanisms by which these states change. So in this example, shrub invasion, or T1, is the transition between a grassland state and a savanna state. And restoration with fire management and shrub control can transition the savanna state back to a grassland. And we also see T2, which is a loss of grass triggering erosion, which can transition a savanna state to a shrubland. And you'll notice there's no arrow leading away from the shrubland state, indicating that this state is highly resilient to change and that there isn't a known mechanism that can be implemented by land managers to transition back out of this state. So each ecological site has a unique state and transition model to describe the plant community dynamics on that ecological site. And having this information helps us monitor ecosystems and make educated management decisions. So for example, we can use a state and transition model to compare the suitability of different ecological sites and states for wildlife habitat, to predict the effect of disturbances, such as invasive plant establishment after fire, and to evaluate whether management objectives are being met within ecological sites and states. So the Natural Resources Conservation Service is developing ecological site descriptions for the United States, though they are used not just by the NRCS, but across agencies and by agricultural producers. And actually this framework is used not just in the United States, but globally. So the intent is to have continuous coverage of this level of information nationally. And as you can see in this map, some areas today have a high density of available ecological site descriptions, and those are the areas shown in a darker orange. And there are other areas where there are few to no ecological site descriptions currently available, shown in gray and in lighter yellow. So we need methods to develop ecological site descriptions where they're lacking. They can be developed with a deductive approach, where ecological site concepts are hypothesized based on accumulated knowledge and observation, and are then tested with targeted quantitative data. And they can also be developed with an inductive approach, where the concepts themselves come from quantitative data. And both of these approaches have been used successfully. An inductive approach allows for ecological site concepts that are grounded in quantitative measurements, which can mean that they are less biased. However, collecting plant and soil data of a quantitative nature across a large landscape is quite resource and time intensive. And federal agencies have been collecting inventory and monitoring data on public and private lands with a suite of standardized methods, building a robust quantitative plant and soil data set. So this presents an opportunity to use this data set for ecological site concept development with an inductive approach, but necessitates the development of a workflow that can be generalized across landscapes, and that streamlines the application of these data to ecological site development. So the goal of this project was to develop a workflow for using existing monitoring data to develop ecological site concepts and to test the workflow in the Rio Grande del Norte National Monument in northern New Mexico. And more specifically, our research objectives were to use the monitoring data to identify ecological site concepts for the monument, to relate vegetation treatments and land use history to state changes, and to produce a workflow that could be generalized across landscapes where there is existing monitoring data and the need for ecological site descriptions. So move on now to the Rio Grande del Norte National Monument. It is one of these areas that is lacking ecological site descriptions. It's approximately 350,000 acres, and it's managed by the Bureau of Land Management. Soils are currently being mapped in this area by NRCS, and ecological site descriptions have been brought in from the Colorado Plateau to be used in this area. They were developed in Utah and Arizona, and there is a large disconnect in the key species and ecological dynamics that they represent and what is on the ground. There are some significant climatic gradients in this area. So it is warmest and wet in the southern part of the monument. It's wettest and colder in the west, and coldest and driest in the north. And there's also an increase in base level elevation moving northward. And representative of these climatic gradients, there are three major land resource areas that converge in the monument. Major land resource areas reflect regional climatic regimes and physiography, and they classify landscapes on a larger scale than ecological site descriptions, but they are part of the same classification hierarchy. And it's important to note that ecological site descriptions are specific to MLRAs and do not cross MLRA boundaries. Here are the major landforms of the monument. So we have volcanic cones. There are over 40 active playas, rolling hills and plains, and then the Rio Grande Rift Gorge, and all of the more specific component landforms that comprise them. The soils in the monument are mostly young alluvial sediments infilled from the adjacent Rocky Mountains. And some of the heterogeneity in soils is clearly related to landform, such as consistently shallow stony soils on the volcanic cones. But some of the soil heterogeneity, particularly of depth and rock fragment content, is not related to landform, but largely due to volcanic vents throughout the monument, which have introduced thin fractured layers of basalt striated at various depths with the soil. In the main vegetation communities we see are mixed conifer and pinyon juniper forest on the cones, a grass shrub matrix on the plains, which is dominated by Wyoming big sagebrush in the south and by winter fat moving north. And there's a smaller area in the northwestern monument dominated by black sagebrush. And it is a multi-use landscape. So today the most important commercial uses are livestock grazing, wood harvest, and big game hunting. And we know from General Land Office records that livestock grazing has been ongoing to some degree in this area since the mid-1800s. And the monument also provides critical habitat to a wide diversity of wildlife species. And actually, 2 wilderness areas were designated in the monument just since the inception of this project, with another currently in legislation, and largely to protect important wildlife corridors. And there are 23 known treatments in the monument recorded since 2002. So these are areas where land managers have initiated a disturbance to remove woody plants, either trees or shrubs, to increase herbaceous cover. And there's visual evidence of additional earlier treatments that we don't have records for. So all of these facets of the monument that I've just introduced, the climate, landforms, soils, vegetation, land use and management history, all play a part in developing ecological site descriptions. So I'll move now to our data set. We use data from the Bureau of Land Management's Assessment, Inventory, and Monitoring Program and the Natural Resource Conservation Services Landscape Monitoring Framework Program. And these are just two examples of the standardized multi-use monitoring programs that have collectively accumulated data from over 60,000 locations in the United States. These programs collect plant and soil data using standardized protocols, and they also use spatially balanced random sampling designs, which means that the data can be aggregated and analyzed at multiple spatial scales. So standardized methods that are core to this, all of these programs are the description of a soil pit to 70 centimeters and the hand texturing of soil horizons. The line point intercept method, which collects plant foliar cover by species, as well as percent litter, and records the soil surface. Vegetation height is recorded for herbaceous and woody plants. Canopy gap intercept records the proportion of the plot with gaps between plant cover. Soil aggregate stability, which looks at the stability of surface soil when exposed to repeated wetting. And species inventory, so all species within the plot are recorded. And additionally, the plots collect information on landscape position and photo points. And all of these methods are described in the monitoring manual for grassland, shrubland, and savanna ecosystems. There are over 200 of these plots in the monument, collected between 2011 and 2018. This workflow describes how ecological sites are often developed. Observational and qualitative data are collected in the earliest phases, and they're used to develop ecological site concepts. And those concepts are then tested with quantitative data collected in the field. So this is a deductive approach, as I mentioned before. And there are many examples of inductive approaches to developing ecological site concepts and state and transition models, with these just being a few. And to reiterate, what makes these approaches inductive rather than deductive is that instead of using data to test concepts that were developed a priori, data are used in an exploratory way to develop concepts from patterns and relationships detected in the data. So building off of these approaches, we developed the workflow that you see on the right. And along with some variation in the methods that were used, what really makes this approach unique is that it was designed to use existing monitoring data as an input rather than data collected specifically for the purpose of developing ecological site descriptions. Both previous inductive approaches and ours use multivariate exploratory analysis to reveal patterns in the data. And typically, multivariate statistics are non-parametric methods used for exploring data sets that are complex and contain many variables. So I'll look at a few of these methods that were key to our workflow in a little bit more depth. So ordination arranges objects spatially based on their similarity across multiple variables, and it plots similar objects closer together and dissimilar objects farther apart. And cluster analysis is used to group objects together based on their similarity. Ordination and cluster analysis are often combined, and when they are combined, they can reveal natural groups in a data set and then visualize how the groups relate to one another. To give you a more visual example, here are unorganized objects, or pieces of candy, and you can see that they're different and similar in multiple ways. And here are the same objects having undergone ordination and cluster analysis. So they were clustered into 15 groups based on similarity, and then the ordination arranged these groups based on their size, which we see on the y-axis, and how tasty they are on the x-axis. So we hope to do something similar with the monitoring data. Of course, these monitoring plots are much more complicated, and each is associated with potentially over 300 variables. And there's a lot of these plots. So it becomes really important to figure out how to reduce the complexity of this data set to reveal the relationships and patterns that we're interested in. The first step of this workflow is to gather and assimilate the data. So we pulled data in from the monitoring plots, from a digital elevation model, climatic data, and predicted soil chemical and physical properties from the digital soil mapping product soil grids. And additionally, some covariates from the monitoring plot soil data, such as depth to maximum clay and available water holding capacity, were calculated, and some covariates from the digital elevation model, such as heat load index and topographic wetness index. And part of preparing the data was to determine how to use foliar cover from line point intercept. So grouping plant species by characteristics such as whether they are annuals or perennials, or shrubs and grasses, and how they reproduce is a way of looking at plant dynamics more mechanistically, and it can reduce potential noise from species misidentifications. So I tried many grouping schemas from very broad classes like tree, shrub, perennial grass, to leaving the species totally ungrouped. And what worked best in the end was a schema wherein the species were grouped by genus and then by functional traits within genera. And perennial grasses were further grouped by photosynthetic pathway and reproductive structure. And there were a few exceptions to this schema. So native, annual, and biennial grasses and forbs were classified together as short-lived native plants. Invasive plants were grouped on their own. And woody plants, trees, and shrubs that had greater than 200% foliar cover throughout the study area were left ungrouped. And this, leaving the woody plants that are predominant ungrouped, was to maintain some important distinctions between shrub and tree species within genera that I hypothesized might be able to provide insight into some of the climatic gradients in this area. So this grouping schema reduced over 300 species to 97 groups. And foliar cover of functional groups was combined with percent bare ground and proportion of the plot with canopy gaps greater than 100 centimeters to represent some attributes of plot structure. So these indicators can provide insight into the potential for wind and water erosion, as well as how resources are distributed at the plot level. And we wanted to use these indicators of plot structure and foliar cover of functional groups to reveal the vegetation communities in this data set. We did that with a fuzzy cluster analysis and a principal coordinates ordination. So the ordination was used really as a venue in which to look at the clusters. So in this graphic, you can ignore the axes. They represent the coordinate locations of each plot. And the colored wedges represent the monitoring plots. So the color indicates what cluster the plot belongs to. And as you can see, most wedges of the same color are clustered in space because they are more similar. And fuzzy clustering is unique as a clustering algorithm because it allows plots to belong to multiple clusters rather than classic crisp clustering, which would be hierarchical clustering, divisive clustering, and k-means partitioning. And those methods force every plot to belong to a single cluster. So when we see a plot represented by multiple wedges, like the ones circled here, this means that they have an affinity for more than one cluster. And the algorithm gives a membership value for each cluster to every plot. So if a plot belongs clearly to a cluster, its membership value will be close to 1 for that cluster and close to 0 for the others. So in this table, we have plot 33 circled there in blue, and we see that it has a high membership value for cluster 7, and correspondingly is represented by a single color. And we also have plot 86 circled in red, and we see that it could belong to cluster 15, and it could belong to cluster 7, or maybe to cluster 1. And correspondingly, it has wedges of multiple colors attached to it. So this method allows plots with ambiguous identities to be flagged and reviewed individually to figure out where they really belong. So I'll return to the fuzzy plots a little bit later. And here are the 16 clusters identified through fuzzy clustering. And they represent vegetation communities defined by functional group abundance and structural attributes. So at this point, I subset the data to only include plots with a membership value to a single cluster greater than 0.6. And I did this so that I could develop modal concepts for the vegetation communities based on just the most clear-cut examples of those communities. So that is what the third column in this table refers to, is how many plots had a high membership value to that cluster. And we'll take a look at some of these modal concepts now. So to develop them, I calculated the minimum, maximum, and mean foliar cover of functional groups and the frequency of occurrence for functional groups from species richness data. So cluster 1 that we see here is driven by big sagebrush and cover of warm season bunch grasses, and 5 plots had a strong affinity for this cluster. So the table that you see is just representing mean percent foliar cover for the functional groups that occurred on all 5 of these plots, to simplify the tables a little bit. Cluster 2 was also driven by big sagebrush, as well as pinyon juniper trees. And cluster 3 was driven by big sagebrush with an increase in bare soil and large canopy gaps. So putting these summaries together, I was able to compare the modal concepts with one another. So in this example, we see big sagebrush driving all of these clusters with mean foliar cover ranging from 23% to 36%. We see 27% cover of warm season bunch grasses in cluster 1, much reduced in clusters 2 and 3. And we see bare soil increase in cluster 3, while cluster 2 has lower bare soil and a higher proportion of large canopy gaps. And I'll show you just a few other vegetation communities. We don't need to worry really about the tables, just so you can lay your eyes on some of the other groups. So this community was driven by winter fat and warm season bunch grass. This was one of the forested communities with a closed canopy and high cover of pinyon trees, an invaded grassland, and perennial bunchgrass community. So we have these 16 vegetation communities defined, and now want to understand where on the landscape they occur and how they might be constrained by abiotic variables. So I first used an analysis called BIOENV, which relates a plant community dissimilarity matrix and abiotic variables. And it first produces a dissimilarity matrix for every possible subset of abiotic variables. And then it calculates the correlations between the plant community dissimilarity matrix and each matrix of the abiotic variables. And it spits out the subset of abiotic variables that produced a matrix with the strongest correlation to the plant abundance matrix. So using climatic and topographic data and monitoring plot soil data, I didn't get above a correlation of 0.32. This is why we included the soil chemical and physical property predictions from the digital soil mapping product, Soil Grids. The Soil Grids predicts soil properties at a resolution of 100 meters and at 6 standard depths. And including these data did increase the correlation to 0.51. So the variables highlighted in red are the ones that BIOENV identified as producing the strongest correlation with the plant community dissimilarity matrix. And soil bulk density was also included in the BIOENV analysis and was selected, but it was removed from further analysis due to some concerns over its accuracy. And additionally, it is what's known as a dynamic soil property. So changes potentially as plant structure and function changes. So useful for understanding ecological states, but not so much for identifying ecological site concepts. So I took these variables produced by BIOENV and used them for a classification tree analysis. So I know this graphic has a lot of information on it, but I'm really just showing it so you can see the overall structure of the tree. Classification and analysis takes a response variable, which in our case was vegetation community, and predictor variables, which were the abiotic variables identified through BIOENV. And the algorithm uses homogeneity within the response variable to split the data set based on values of the predictor variable that best predict the response or vegetation community. And we pruned this tree so that it had 12 splits and produced 13 ecological site concepts. So as an example, we'll look at just a few branches of the tree. We started with 139 plots that comprise the modal concepts of the vegetation communities. And we see that the first and most predictive split is on soil pH at 30 centimeters. That variable comes back for the next split, and then soil depth and elevation and slope and pH again. So I used these breaks identified by the classification tree to develop ecological site concepts. These are 6 of the 13. So although landform wasn't included in the classification tree analysis, I was able to map the plots that were classified into each of these concepts and look at the relationships that they had with landforms. And each of the 13 ecological site concepts is associated with a set of vegetation communities. So this table represents the 13 concepts. And every plot in our data set was assigned to one of these concepts based on the quantitative values produced by the classification tree. So another way of understanding the distribution of vegetation communities over abiotic variables is to visualize them in box and whisker plots. And I'm not showing these figures to highlight any particular inferences, but really just to highlight this phase of the workflow and show that we can see how the vegetation communities are similar or vary in both the variables that we used in our classification analysis. So the two on the left, elevation and annual precipitation, as well as looking at other variables that were not part of the classification tree, but that often influence ecological sites, such as percent clay at the soil surface and rock fragments. So looking at differences in the ranges of these variables across the vegetation clusters can show which communities might belong to different ecological sites where they don't overlap, or which communities might belong to multiple ecological sites, or an ecological site with a large range of variability if they occupy a large range, or if they consistently occur over small ranges of these variables, they may be constrained to a single ecological site. And again, I don't mean to overwhelm with these plots. I know there's a lot of detail there, but this is just to show the method by which we can compare differences among these vegetation communities. So we can also look at indicators of ecosystem structure and function that were not included previously in the analysis, like height of woody and herbaceous plants, percent woody and herbaceous litter, and soil stability classes. And now we'll come back to the fuzzy plots, those plots that had a membership value of less than 0.6 to a cluster. They were reviewed individually, and I looked at their highest membership values, the modal concepts to which they had the highest membership value, and how much they differed from them, whether it occurred in a treatment, where on the landscape it was, and what it looked like in pictures, what ecological site concept it matched, and whether it was different enough from the existing modal concept that it warranted the development of a new class. These are the 5 additional vegetation communities that were represented by fuzzy plots, where I felt that the fuzzy plots were too different from the modal concept to be grouped together. And these 2 rock outcrop communities were also used to form a 14th ecological site concept. So now all of the plots, both those that formed the modal concepts and the fuzzy plots, have been classified to ecological site concepts. And the vegetation communities that occur on each of these concepts are summarized. So you see that in the central column of this table. And I also looked at the major land resource areas that each ecological site concept occurred in. So 4 concepts were constrained by a single MLRA. 3 concepts were mostly constrained by a single MLRA, but had 1 or 2 plots close to a boundary, in which case I think they probably are confined to a single climatic and physiographic region, but that MLRA boundary might not be perfectly drawn. And then there were 7 sites that had multiple plots in multiple MLRAs. So these, I suspect, are actually comprised of multiple ecological sites that we didn't have a variable to that was able to differentiate among them. Here's a more graphical representation of how the vegetation communities are distributed across an ecological site concept. So this is an ecological site concept with low slope in the eastern part of the monument in the lowlands with a high pH that occurs in MLRA 51. And we see on this site a vegetation community dominated by perennial bunchgrass and subshrub, one dominated by grass, shrubs, subshrubs and large gaps, winterfat and perennial grass, and winterfat and bare soil. And I recalculated the mean values of functional group cover based just off of these plots in this ecological site concept. And when grouping the vegetation communities within ecological site concepts, I stratified them by treatment when there was a known treatment there. So in this example, one community represented plots that had been treated mechanically. And in this example, the ecological site concept was associated with 3 vegetation communities where there was no treatment and 3 vegetation communities where there was a mechanical treatment. And the big sagebrush bare soil community occurred both in treated and untreated areas. There weren't enough plots within ecological site concepts and treatments to use another classification tree analysis to predict how plots might respond to treatment based on ecological site. So these relationships that we identified between ecological sites and treatment history are preliminary, and we will collect additional data to verify those relationships and to be able to turn the assemblages of communities within an ecological site into more of a state in transition model. And I took this table that we have seen before of ecological site concepts and vegetation communities and turned it into a dichotomous key that groups every plot into its ecological site concept and treatment type. So we saw 3 ecological site concepts with 6 vegetation communities on the volcanic cones, 9 concepts for ecological sites with 20 vegetation communities in the plains and low hills, and 1 ecological site concept with 4 vegetation communities that occur just on the rim gorge. The concepts developed by this workflow will now be verified with additional data collection and through workshops with land managers and stakeholders. Data collection will focus on gathering more information for the vegetation communities that were represented by just a few plots, as well as looking for additional soil and site characteristics that will help match these ecological site concepts to soil series. And we also will want to verify their ranges of variability. So because the monument is an administrative boundary and ecological sites extend beyond the ranges of the monument, we will collect data outside the monument to look at the full extent of these ecological sites and make sure that the concepts represent those full ranges of variability. And we hope that workshops will help to ensure that the resulting ecological site concepts are relevant to land management, as well as providing more insight into additional treatments and land uses that we didn't have quantitative data for. So this workflow can be generalized for other landscapes by including additional data and changing the way that these are grouped. There are a number of parameters involved in fuzzy clustering that can be manipulated. Deciding how the classification tree is pruned, and deciding when a plot is different enough from a modal concept that it warrants the creation of a new group. So to conclude, we identified 14 ecological site concepts and 21 vegetation communities, and these were organized in a dichotomous key. The key was then stratified to represent treatment history when possible, though we'll need additional information to really verify the drivers of state change. And a multivariate workflow was developed for using existing monitoring data to develop ecological site concepts. And in addition to being used to develop ecological site concepts where they don't currently exist, I think that this workflow could be useful to test provisional ecological sites and the ranges of variability that they describe where there is monitoring data. And because each modal concept is associated with quantitative values These values can be used to build management benchmarks, though additional monitoring data would need to be collected to evaluate those benchmarks. So the data wasn't being used to build the model and to compare against it. And as in our study area, this approach might be able to provide insight into broader landscape classification units like MLRAs. And because many of these plots are revisited on a 5-year rotation, this workflow could be expanded to track plots as they transition or don't transition through states. Some future research questions of interest are looking at how monitoring plot soil data might be better used in this analysis. Depth was the only variable from that data set, from the monitoring plot soil pits that emerged as having a strong relationship with our plant data, but perhaps looking at broader textural classes might reveal additional patterns. Looking at how a location-specific digital soil map could improve the workflow, at how ensemble classification tree methods might improve the workflow, how this could be integrated with multispectral remote sensing data, and with existing models that look at things like wind erosion and water erosion potential, how it might interact with the ecological site database edit and the landscape data commons, and how these modal concepts might be used to support the evaluation of ecosystem services across sites and states. So thank you all so much for listening. There are so many people who made this project possible, and it's been a really incredible experience to get to work on this project, and particularly to use this data set that I spent a lot of time collecting and apply it at a higher level. So in particular, I would like to thank my advisor, Nick Webb, for his endless support throughout this process, and Zoe Davidson, who has been such a strong career advocate for me over the years and who really brought this project to fruition. And then of course, my partner and my family, whose support really carried me through this process. So again, thank you all, and I am happy to answer any questions.
Thanks, Ali, for really clear and comprehensive talk. So I've got one question that's come through the chats here from Peggy Brown. What was the rationale for your invasive species grouping, and didn't they possess functional characteristics of other groups?
Yeah, that's a great question. So they did. All of the species that were classified as invasive species were either annual forbs or annual grasses. And I used a species list for New Mexico that was put together by Patrick Alexander for use with the AIM monitoring program as the basis for grouping those invasive plants together.
Fantastic. Does anyone else have any questions for Ali?
Yeah, but I can't type that fast, Nick.
Well, you can. How about you can ask that question?
Thanks, Al. This is Jeff Herrick. That was an amazing presentation. Really well done. Great study. I'm just wondering, the inductive approach, I think this is one of the best examples or best applications, the most effective applications I've seen of an inductive approach in the past. I've been a little bit skeptical. And one of the things I was just wondering, though, as you go back through it, have you thought about why temperature didn't fall out? And I'm just thinking about plant ecophysiology and the altitude range that you've got there. Any thoughts on that?
I wonder if, as we look at the larger ranges of variability for these concepts, if temperature might fall out then. And though there are, as we saw, some pretty strong changes in temperature just in the monument, I think those, where these MLRAs meet, those gradients will become bigger as we look at areas outside of the monument. So that is definitely something that I would want to pay attention to as we try to understand sort of the larger ranges of variability for all of these concepts.
Thanks, Ali. So I have a question from Zoe, which is how easy would it be for land managers and NRCS to use this workflow? Do you think it would require a specialized skill set?
That's a good question. I think certainly familiarity with some of these analytical methods is important, though easy enough to learn with some time and effort. And I think that this workflow could be used by land managers who are familiar with landscapes. And even though it is an inductive approach where the data is, put in and in a sense is then spit out into these classes, there are a lot of steps along the way that rely on a familiarity with the landscape and understanding what is there. So I think in some ways, a land manager who has spent a lot of time on that landscape might be well suited to using this workflow.
I have a follow-up question from Patrick regarding Jeff's question about elevation, wondering if part of the answer might be that elevation and landform are correlated.
I think that is part of it. In this particular landscape, there is a weaker correlation between elevation and temperature than there might be, I think, in other landscapes. And I believe that's because we have these volcanic cones that are higher in elevation and are colder, but that there's also this kind of general increase in elevation and decrease in temperature moving northwards. So some of the cones in the southern part of the monument there may be a higher elevation associated with a warmer temperature in that area compared to the northern part of the monument. So I guess what I mean to say is it's not quite as straightforward of a relationship as perhaps in other landscapes.
I have a question from Dawn Browning, who starts with "Awesome job, Ali," and a follow-up to Jeff's question: were you surprised by the prominence of soil pH being at multiple splits in the classification tree?
I don't know if I was so much surprised by that. I think having worked in this landscape and sort of done just very basic exploration into it, it seems like the accumulation of calcium carbonates in the soil is really one of the driving forces behind these plant communities. I guess I am a little bit surprised that, because I think that's largely what the soil pH variable is picking up on, but I am surprised that some of the covariates I pulled out of the monitoring plot data, like how effervescence classes change throughout the soil pit and the maximum effervescence class, that those weren't identified by the BIOENV routine in addition to soil pH or instead of it.
Excellent. Any other questions from anyone else? Okie dokie. Well, thank you very much, Ali, for a fantastic presentation. We might wrap it up there and move on to your private defence. thank you again, everyone, for joining and listening in. I know that Ali really appreciates it. Yeah, so well done, Ali. Thank you.
Yeah, thank you all.